right. Good afternoon, everybody. I'll go through what you should have done yesterday and what you should be doing today, and give you time to work so that you can prepare for the test that's on Monday. Okay, you can either go through the biology. All right, so you either go through the dashboard or the calendar. I think the calendar is a little quicker. So I go to the agenda. This is for today. Like I said, I'm going to go backwards and look at Wednesday. Yesterday was supposed to be a day that you actually did work on your own. I left you a video going through what you needed to learn how to do or know how to do. I added this here because you will need to know that for the test. If it were me, I'd write it down to remember. But DNA to DNA replication is called semi-conservative replication because we took DNA, unwound it, and made an old strand with a new strand, and that's semi-conservative. Here is a practice for all the things we learned: translation, transcription, replication, all in one spot. So I'm going to show you an example here of what it looks like. If you're supposed to take a dice, I had a student in the class earlier that we used cards to do the rolling for dice. You just select a card 1 through 6 and then make them each numbers, whatever. You're going to come up with your DNA strand. Then you're going to take that DNA strand and make an mRNA sequence. Remember, the difference between the two is the T is a U. Then you're going to take 3 at a time. These 3 will be a codon. The next 3 will be a codon. The next 3 will be a codon. And the next 3 will be a codon. And then you're going to find your protein amino acid when you go back to, let me go backwards, back to this page here. You're going to have this picture here. I'm going to quickly show you how to use that. If you look at this, you're going to see that on this side, we have first position, second position, third position. Kind of think of it like a puzzle. I'm just going to come up with a codon. AUG is usually a start codon to start making that polypeptide chain. The first position says I have an A. So I'm going to look for A. That means I'm looking for anything in this box here. Then I'm going to look for a U in second position, which means I'm looking for anything in this box here. And then the third position, which is a G, which is right here, which means this is what I'm looking for. Because as you continue to narrow down your focus, it leads you to your final answer, which is AUG is MET. MET is the protein that you're going to put down as your answer if you have AUG as your codon. This is your codon. This is your amino acid or protein that you're going to list there on that page. Now, yours may be different. You have to look and see what your code is. I'm going to use the same code over here. This is similar, same sort of thing. As a matter of fact, nobody's saying anything, but I'm pretty sure I'm not sharing with you so you can see it. You guys are going to have to speak up. If this is AUG for this one, I'm going to start in the middle of the circle and select A. Then I'm going to see that this pi piece here is the one I'm talking about. So that would be second position, which is right in here. I'm going to look for U, which is here. Then it's telling me I'm looking inside this small pi piece, and I'm going to look for G. and there's G, and there's my answer. It's MET. Both of these will get you the same answers regardless which one you use. However, this one here, I am told, is on iStep or iLearn. If you go down here to the bottom, it's going to be, oops, that's my, let me take that away. This here is actually my my copy. I did it myself. I came up with my DNA strand, my sequence there by rolling the dice. I took my DNA strand and converted it to mRNA by changing all the A's to U's, not A to T. I then took 3 at a time and put them down here. These are called codons, 3 at a time, 3 at a time, 3 at a time, 3 at a time are a codon. And then I looked it up on that chart, the circle chart or the other chart, and I found my proteins here. And that's how you do that page. This here, if it were me and I were taking the test, I'd have this printed out as an example, ready to use so that I could do well on the test, because this does have a lot of information on it to help. This last thing on yesterday's assignments is an extra credit assignment. You're going to build a monster. And what you're going to do is you're going to do, again, taking dice and rolling. This is worth 30 points of extra credit. You can go through and you're going to find the eyes, the legs, the arms, the hair, so on, so on, so on. And then down here, you're going to roll. You're going to roll the dice or tell what the roll was that you had, tell the DNA sequence, do transcription into the mRNA strand, meaning the A, instead of T's, you're going to use U. You're going to do translation, which tells me which amino acid you have. And then the phenotype is telling me what they look like with your eyes. If you looked at them with your eyes, what would they look like? Then down here, excuse me. Down here is where you're going to draw your monster. And that's how you do that extra credit assignment. Now we're going to go back to the calendar and we're going to look at the assignment for today. We're getting down to the nitty gritty of what we need to know for our test. So I've given you a video over mutations. These 3 mutations right here, deletion, insertion, and substitution, are the 3 that you have to know. Deletion means it takes a base out. Insertion means it puts a base in, and substitution means it replaces one. So when you look at a DNA sequence, you should be able to see which one doesn't belong by knowing whether it's deletion, insertion, or substitution. The last thing on here is just some examples of different ways to look at the mutations. Here is a practice game. It'll take you through, you make your DNA strand, then you make an RNA strand, and then you make a protein. It kind of goes through the whole process of protein synthesis of what we've talked about. There's even a portion on here where you can do mutations as practice. This next part here is a quick little go through reminder of mitosis, which is something you should have learned in middle school. We're just going to review it. It is on the test. We'll look at the study guide here in a second. So in interphase, what happens is the chromosomes and the nucleus are going to get loose like spaghetti so that they can be copied. So you're going to see that we're going to copy the DNA. You're going to have your centromeres up here, or centrioles up here. Then as you move down to prophase, this is the longest cycle up here. Interphase is the longest cycle because it's growing. It's growing in chromosomes. It's growing and getting ready to make copies of the whole thing. So interphase is the longest phase. Prophase, you're going to see the nuclear envelope start to disappear. The centrioles moving to the opposite edges of the cell, which are going to be called the north and south pole. These are the poles. So these are the poles. They get to the cross the poles in metaphase, and the chromosomes line up in the middle. That's why it's metaphase middle. The spindle fibers, these long lines, are spindle fibers, and they're going to attach to the centromere in the middle of the chromosome. In anaphase, what happens is that centromere hooked onto the spindle fibers will start to pull towards the poles, making the cell longer. So it's getting to be like a long, like egg shape. Then it draws those chromatids. That's what those half chromosomes are called, chromatids. And then in telophase, it kind of looks like a peanut. The peanut's starting to show that the cells are pulling apart. You're going to have the chromosomes divided into the cells, or the chromatids that were pulled to the sides. You're going to have elongation of the cell continuing to happen. You're going to have the nuclear envelope starting to reform around the cells. And then in the end, you have cytokinesis. Cytokinesis just means to split, is what cytokinesis means. It splits. And then again, the cells go back into looking like spaghetti and all over again. And this is what typical cells do. Cells, any cell in your body does this. The other type of cell division is for sexual reproduction, and that's not what this is. Okay. So the last thing on here is a study guide. I gave you a study guide yesterday if you looked, and I'm replacing it with this one. I thought I'd give you two, but now I've decided just this one will do. But if you look here, there is a spot with cell division in it. I would suggest you know this and have it filled out. If it were me, I'd print this off, have it next to me while I take the test, and make sure that I'm using it to my advantage. The test is timed, so you don't have all day to get it done. The test is on Monday, but you are timed, so I make sure you know enough to be able to get through the test and, you know, do your best. Are there any questions? All right, if you don't have any questions, you're free to go.
Have a nice day.
You too, take care.
Have a nice day.
You too.